All right, welcome everybody. My name is Hunter, and this is week 4 for the Learning Center Principles of Biology review. Today we're covering viruses, bacteria, and archaea. So how do viruses affect host cells? So by definition, viruses are obligate intracellular parasites. Obligate intracellular parasites. What does this mean? We're typing today. I'm gonna forego the stylus today. If we just look at the vocabulary here, obligate, obligation, we've heard these words before. Obligate means it's necessary for something. Intracellular, prefix intra, inside, cellular, the cell, so inside the cell. So it's obligatory that something occurs inside the cell. Obligate intracellular parasites. Parasites feed on other organisms. So viruses, in order to survive, necessarily must parasitize or feed on the inside of other cells. More precisely, an obligate intracellular parasite is one that can neither survive long-term nor reproduce outside its host. So very often when we're talking about viruses, we'll use language that is very similar to how we talk about other living organisms. Viruses are not technically living organisms because of this trait. They necessarily cannot reproduce on their own. They're basically protein packets of DNA information which interact with the machinery of other organisms that facilitate reproduction of the viral DNA. So, in general, how are viruses classified? Kind of like I was alluding to a moment ago. Viruses are classified based on the type of nucleic acid that they contain. So for example, single or double-stranded RNA or single or double-stranded DNA. So viruses are classified based on the type of nucleic acid that they contain. For example, single or double-stranded RNA or DNA. Single or double-stranded RNA, single or double-stranded DNA. So what is reverse transcriptase? Where was reverse transcriptase first found? Of what use is it to viruses? Of what use is it to scientists? So this is a useful factoid, but it's also going to be, it's a cool little factoid, and it's useful in that we can thinking back to our previous conversations about bacteria and how we might choose drugs to treat different kinds of bacterial infections, it's useful to understand machinery that is unique to viruses, not just because it's good to understand, but because it gives us targets for treatment. So reverse transcriptase is one of those features. Reverse transcriptase was discovered in retroviruses, which is any of a group of RNA viruses which insert a DNA copy of their genome into the host cell in order to replicate. And the most famous example of these is HIV, which we've talked about before. Reverse transcriptase, which I'm just going to abbreviate here, you probably should not do that in your homework, but I will, is the enzyme that allows retroviruses to make DNA from RNA, from RNA strands, and then I think this abbreviation is add or subtract RNA from the DNA. It's useful in genetic recombination to make cDNA from mRNA and avoid the problem of eukaryotic introns. Introns, again, are those portions of DNA that don't get expressed in the genome. So it's useful in genetic recombination to make cDNA from mRNA, messenger RNA, and avoid the problem of eukaryotic introns. So if it spliced itself into a eukaryotic intron, it wouldn't get expressed. This enzyme sort of bypasses that consequence, is my understanding of it. So why must viruses invade other cells to reproduce? We've mentioned that this is necessary. It's obligate intracellular parasite. But why? Why is this necessary? Describe the general process of virus reproduction and include discussion of the lytic and lysogenic virus cycles. So I'm going to see if I can just expand my window here so you can see this graphic. This is a graphic from the Pearson textbook that I found really, really helpful in describing the virus life cycle. This is very flashcardable. It's just good to have a solid understanding of what's happening here. So we have the lytic cycle, in which the host cell lyses, breaks apart as part of the virus reproductive cycle. And then we have the lysogenic cycle, which is this recycling really of the prophage, the viral DNA, into and out of the host cell's chromosomes. So grab a screenshot of that if you want, turn it into flashcards, and this will kind of be a helpful reference for us as we're talking about this. So describe the general process of virus reproduction, include a discussion of lytic and lysogenic virus cycles. All right, so some viruses are only lytic. They enter the cell, take over, the cell's machinery to make more viruses. Then they lyse the cell to allow the new viruses to escape and attack other cells. Some viruses also have a lysogenic stage, and this is and alternate between lysogenic and lytic stages throughout their life cycles. Some also have lysogenic stages and alternate between lytic and lysogenic cycles throughout their life cycle. In the lysogenic stage, the virus incorporates the viral DNA into the host DNA and remains quiescent there, replicating with the host's DNA. You might hear my neighbors here, sorry about that. In the lysogenic stage, the virus incorporates the viral DNA into the host DNA and remains quiescent. Ooh. Cool. So then it can later come out of the host's DNA and enter a lytic cycle to then infect other cells. So environmental conditions that stress the host cell may trigger this process. All right. Part B, which type of viruses are more likely to have a lysogenic phase? So this is going back to that classification question. So there are a number of double-stranded DNA viruses and retroviruses that can produce double-stranded DNA and can become lysogenic and incorporate into the host DNA. So a number of double-stranded DNA viruses and retroviruses that can produce double-stranded DNA. A number of double-stranded DNA viruses and retroviruses that can produce double-stranded DNA, can become lysogenic. RNA viruses that do not produce DNA as templates for their reproduction are unable to incorporate into the host DNA. So RNA viruses cannot be lysogenic. So that's a simple thing to have memorized. If a virus is incorporating its DNA into the cell's chromosomes, into the cell's DNA, then it is lysogenic. But if it's an RNA virus, it by definition cannot incorporate its DNA into that genome. All right, so more than 100 different viruses, many of them rhinoviruses, can cause the common cold in humans. Other viruses, influenza viruses, cause the flu. And although there are many different antibiotics for treating bacterial infections, relatively few drugs are available to treat viral infections. Explain. So antibiotics are useful to counteract bacterial infections because they target bacteria-specific structures or functions. Let's go ahead and write that down. Antibiotics used to counteract bacterial infections target bacteria-specific structures or functions. These might include the 70S ribosomes, which are unique to prokaryotes, bacterial cell wall structures, bacteria-specific enzymes. So anything that is unique to bacteria and not part of eukaryotic cell infrastructure make good targets, especially if they facilitate reproduction or mobility or the physical structure of the bacterial cell. So it's a lot more difficult to find something that a virus requires for its function and replication that is not also required by the host for its normal metabolic functions. So let's go ahead and write that down. It is much more difficult. It's much more difficult to find something that a virus requires for its function and replication that is not also required by the host for its normal metabolic function. This is because it hijacks the host cell's machinery for its own reproduction. So when a virus-specific coat protein or enzyme is found, for example, reverse transcriptase, high rates of mutation often make it difficult to develop a drug or vaccine. So even in the case where we find a virus-specific coat protein, the high rate of mutation can still make it difficult. So when a virus-specific coat protein or enzyme is found, such as reverse transcriptase, so the COVID mRNA vaccines are a really fantastic, salient example of a viral structure that was highly conserved meaning it changed very little from generation to generation in the cells, in the virus's life cycle, becoming the target for a vaccination. Scientists developed a messenger RNA vaccine that basically prompts your cells to create that extracellular protein, so that your immune cells could recognize it and then build a defense against it without having to be infected with COVID. Let's see. Let me make sure I got everything on this point before we move on. of note for this, reverse transcriptase has no proofreading capacity, and that's part of why mutations are so much more rapid in viral DNA than in eukaryotic DNA. Eukaryotic DNA, there's proofreading as part of the replication machinery that's happening. DNA ligase and a few other enzymes. All right. So let's develop a drug combination to effectively combat multiple infections. Patients with AIDS often acquire opportunistic infections. Opportunistic meaning immune system was suppressed by the initial infection and then something else took hold. Imagine a patient with AIDS and 2 opportunistic infections, a respiratory disease caused by Mycobacterium and another disease caused by the fungus Candida. As the patient's physician, you need to prescribe drugs to counteract the infections. Given the normal anatomical and functional characteristics of each disease organism, what characteristics should a drug combination have to treat each of these 3 infections while doing the least harm to the patient? We have a very small box to type in here, and I have a ton of notes because there are a lot of possible answers here. And this, if you want to pause, if you're watching this in the future and you want to pause the video and go work on this, do it. It's a great exercise. Just find out everything you can about the AIDS virus, about the virus that causes AIDS, about Mycobacterium, about Candida. These are very common infections, very well known and well understood infections, and so there's a lot of information out there about them. But the main things to keep in mind here, there are 2 main points. The treatment needs to address each of the 3 infections separately, and treatments would need to attack the unique characteristics of each of the disease organisms. So for the AIDS virus, for example, the patient will need continual treatment using several different kinds of drugs because the virus will eventually adapt or evolve to those treatments. So these treatments could inhibit proteins on the virus that link to receptors in the cell, much like the mRNA vaccine for COVID works, allowing it to enter or leave the cell. Alternatively, the drug may inhibit reverse transcriptase and prevent the virus from making DNA from its RNA and therefore from replicating. And in reality, treatment of AIDS is very difficult because its DNA can integrate into human chromosomes and lie dormant and hidden for long periods of time. In addition, the virus that causes AIDS undergoes very rapid evolution via mutation. So as existing drugs destroy the virus mutants that are susceptible, those that are not susceptible can continue to infect. And so it's going to be a field of study that's around probably for as long as there are humans, because the organisms are adapting as faster than we can come up with treatments for them. So drugs that target viral machinery must be constantly developed as the virus adapts or mutates. Mycobacterium. So we kind of mentioned earlier, best option for treating bacterial infections is an antibiotic that targets a specific bacterial trait not found in humans, such as a cell wall or the 70S ribosome or something similar. So Mycobacterium is not affected by something like penicillin, which compromises the peptidoglycan wall in the cell wall of many bacteria. So because Mycobacterium is not affected by penicillin, we would have to use something else. So drugs like erythromycin, which interfere with bacterial protein synthesis by affecting the 70S ribosome of bacteria, which would differ from the 80S ribosome in eukaryotes. Erythromycin. And finally, Candida. So Candida is a fungal infection. Sorry outside of our box. That's fine. So this is a little more complicated. Antibiotics aren't going to work because yeast cells, candida cells, are eukaryotic, like human cells. So given the many similarities in eukaryotic cell structure and function, the fungal infection is much more difficult to treat than the bacterial infection. However, it may be possible to target the fungal cell walls because humans have no cell walls to damage. It may also be possible to destroy the fungus exoenzymes, outside enzymes, that are needed to break down nutrients. So there are antifungal treatments. Cell wall or exoenzymes. Cool. If you've ever seen athlete's foot cream, that's specifically for treating tinea. Tinea is a very common fungal infection. It's a skin infection. We've talked about it before, ringworm, right? So named for the ring-shaped rash that it forms as it eats its way out from the center. The common drugs for that are clotrimazole and terbinafine, and a few others, but those are the over-the-counter ones that you can get. All right, so what are the main characteristics of bacteria and archaea? So based on DNA sequence analysis, 3 major domains of life have been proposed. What are the 3 major domains of life? What sets of characteristics place organisms into one domain versus the other? All right, so the major domains, it's in the header. All right, we've got bacteria, we've got archaea, and we have eukarya. We belong in eukarya. Bacteria is a lot of what we've been talking about, and archaea is kind of unique. We're gonna break that down a little bit. So key characteristics of bacteria. Both bacteria and archaea display the prokaryote cell structure. Prokaryotes, remember, there's no nucleus, is a major tell. Let's see. Most bacteria have a circular chromosome. They have cell walls with peptidoglycan and muramic acid. Muramic acid occurs naturally as N-acetylmuramic acid in peptidoglycan. So all bacterial species use the same type of RNA polymerase. They use formal methionine as their initiator amino acid to start protein synthesis. Formal methionine. A formal group is added to the amino group. It is specifically used for initiation of protein synthesis from bacterial and organellar genes. I really wish I could. There. All right. So those are the key characteristics of bacteria. Let's move on to archaea. Archaeal cell walls do not contain peptidoglycan, or muramic acid. Like bacteria, they also have circular chromosomes. They have several types of RNA polymerase. Where several types of RNA polymerase can be found among different archaeal species. And archaea use methionine as the initiator amino acid in translation as opposed to formal methionine, formal group in bacteria. All right. Eukarya, eukaryotes. Humans, funguses, birds, fish, plants, everything. Eukaryotes have the eukaryotic cell structure, which includes double membranes, double membrane-bound nucleus and membrane-bound organelles. Eukaryotes have linear chromosomes. Linear chromosomes. I need some caffeine, y'all. It is so nice and sunny today, and I want so badly to be outside. But I'm happy to be here doing this review, and afterwards I'm going to treat myself. I'm going to go blow off my my midterm study for a half hour. I'm going to ride my motorcycle. It's going to be great. All right. Linear chromosomes in eukaryotes. Eukaryotic cell walls, when present, for example in plant cells, generally contain cellulose or chitin. All right. Eukaryotic cell walls, when present, i.e. in plants, generally contain cellulose, chitin. Eukaryotic methionine is the initiator amino acid in translation. Translation. There we go. So note some similarities between eukarya and archaea. Methionine is the initiator amino acid, and that's the big one, right? All right. Bacteria are divided into 6 major groups. What characteristics are used to place organisms into each of the groups? All right, firmicutes. Firmicutus. If you've ever heard the term cuticle, you've got a hint here. Firm is exactly what it looks like. So this is all very Latin derived, I think. Firm literally means hard, and cutes could be, in this case it's referring to the cell wall, so we can think of it as like a skin. So firmicutes are gram-positive, which is another way of expressing this, big cell wall. DNA contain low percent guanine and cytosine. They have rod or sphere-shaped, whoa, we're running off the page. Sphere or rod-shaped. Some can fix nitrogen. Some can photosynthesize. Some metabolize via fermentation. So yeast. Some also metabolize through cell respiration. We got both. Members of this group cause anthrax. These are examples again. Botulism. If you've ever eaten expired canned foods or canned foods with a crack in the seal, it's a good way to get botulism. Tetanus. And some are used to ferment milk products. So we think of these things often in terms of good and bad, useful or not useful, but it's rarely that simple. Spiracales. Characteristic corkscrew shape. They metabolize via fermentation. Running off the page again. Fermentation. Some can fix nitrogen. Examples of spiracales include syphilis and Lyme disease. And many are anaerobic. Anaerobic, meaning they do not require oxygen for metabolism. What's happening here? All right. Actinobacteria. Actinobacteria have DNA with a high percentage of guanine and cytosine. All right. These are gram-positive, like our firmicutes. Rod and filamentous shapes. Heterotrophic. Hetero, other, trophic, consumption. So heterotrophic meaning they consume other organisms for food. And parasitic. Yep. Source of many antibiotics. Also the cause of tuberculosis. And leprosy. Interestingly, tuberculosis is one of the... I mean, I actually looked this up. Worldwide, tuberculosis, or TB, is the 13th leading cause of death and the second leading infectious killer only after COVID-19. And we have vaccinations against it, I'm pretty sure. Don't we? Wait. I'm going to eat my words here. Yeah, BCG is a vaccine for TB. Vaccine is not widely used in the United States, but it's often given to infants and small children in other countries where TB is more prevalent. Places where tuberculosis infections are more prevalent in the United States include prisons. So if you work in hospitals and prisons, you have to get tested for it fairly frequently. Sorry for the tangent. Keeps it interesting for me. Hopefully it keeps it interesting for you too. So actinobacteria, known to cause tuberculosis and leprosy, and some are abundant soil decomposers. All right, chlamydiales. These are unique because they are spherical shape and very small. All known species are parasitic and live inside host cells. Causes the STI chlamydia. And it's the most common cause of blindness in humans. All right, cyanobacteria. Cyan, literally the color. So named because of their color. So cyanobacteria are cool because they can have solitary or colonial forms. They are the dominant mass of aquatic environments. Meaning they make up more biomass than any other group of organisms in the ocean. All are photosynthetic. Some can fix nitrogen. Some live in association with fungi called lichens. Cyanobacteria is really, 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 really neat. I had the privilege of taking a historical geology class for 2 blocks, 8 weeks, at Colorado College in Colorado Springs. And for those entire 8 weeks, we were doing field trips all over the western U.S. And we got to go and see the stromatolite structures. And these were geologists believe fossil structures of cyanobacteria colonies. I'm pretty sure. Let me... Stromatolites. Stromatolites are bizarre fossils whose biological origins were debated until only a few decades ago. Today, scientists generally agree that stromatolites are layered colonial structures predominantly formed by cyanobacteria. That flashcard hasn't come up in my queue in a very long time. I'm gonna take that win. Yeah, so cyanobacteria are really, really important early organisms in Earth's history and sort of predate most other plants. Well, they predate plants and probably gave rise to photosynthetic plants. Dang it, that was a cool class. I haven't thought about that in about 8 years. Yeah, really, really cool stuff. All right. Proteobacteria. Proteobacteria has 5 major subgroups. We have rod, sphere, spiral shape. Oh, I'm sorry. I guess we're not naming the 5 subgroups. They come in rod, sphere, or spiral shapes, have a wide metabolic diversity. Can cause legionnaires. Actually want to look up. I don't actually know a lot about Legionnaire's disease. Legionnaire's disease is a serious type of pneumonia, lung infection caused by Legionella bacteria. People can get sick when they breathe in small droplets of water or accidentally swallow water containing Legionella into the lungs. There you go. So Legionella is a proteobacteria that causes this infection. So it can cause Legionnaire's disease, cholera, dysentery, gonorrhea, ulcers, diarrhea. So everything from respiratory to gastrointestinal to STIs. They're also used to produce vinegar. Again, good and bad, helpful and not. The genus Rhizobium lives in or on plant roots and fixed nitrogen. Yeah, so remember the mycorrhiza, proteobacteria that live in association with plant roots, increase their surface area, increase nutrient uptake, also fix nitrogen into the soil and make it more accessible for plants since they cannot get it directly. All right. Time check. We're at 4:39. We've got 20 more minutes, and we're on our last page, so I think this will work out just fine. Chats are quiet today, which is fine. I understand that 4 p.m. on a Thursday is kind of an inconvenient time for folks. Unfortunately, it's what worked for me. So I appreciate those of you that are able to sign in, and for everyone else, I hope you enjoy the recorded sessions on YouTube. All right. What kinds of arguments or evidence would you use to support the idea that bacteria are more highly evolved? So big old disclaimer here. Answers to this question depend a great deal on how one defines evolved. Many kinds of comparisons can be made. We're going to provide one example below. Here's one example. All right. If we consider organisms that are closely fit in their metabolism, for example, to their environment, we could say that it is more highly evolved. So then many of the species of bacteria are more highly evolved than humans in that sense. So if we consider organisms that are closely fit in metabolism to their environment as more highly evolved, then we could say that many of the species, many species of bacteria are more highly evolved than humans. So we're not, in this case, talking about degrees of complexity. We are talking about success within a niche, right? Fitness in terms of ability to pass on your DNA to the next generation. How would you support the idea that humans are more highly involved? So for me, my brain jumped immediately due to degrees of complexity. So if we use the term evolved to indicate major changes in morphology, the physical makeup of an organism, that allow organisms to survive in a number of different habitats, then humans are more highly evolved. So if we use the term evolved to indicate major changes to morphology that allow organisms to survive in a number of environments or habitats, then humans are more highly evolved. So bacteria are really specialized in their specific niches. We could say that that is a higher degree of evolution. Humans are more adaptable to various environments within certain parameters. We could say that then humans are more highly evolved. All right, so given what you know about the origin of life on Earth, you want to look for life on other planets. What characteristics of the planet's environment would you look for? And explain your reasoning. And again, science fiction is a really fantastic source of inspiration here. The best science fiction is grounded in a really solid understanding of the principles that you are all learning. So if you do not go on to become scientists, please go on to become storytellers who can tell convincing science fiction stories. They're my favorite kind. The Martian comes to mind again. What characteristics of the planet's environment would you look for? Explain your reasoning. All right. So although we know that life can exist in the absence of oxygen, to the best of our knowledge, it cannot exist in the absence of water. So water is a big one. Therefore, one of the first tests would be to test for the presence of free water. And I believe in my lifetime, we have gone from being pretty sure that there was no water in our solar system to being pretty sure that there is water on Mars and there is water on Europa. So that's exciting. That's just a stone's throw across the galaxy. That's pretty close. What kinds of life would you look for, and explain your reasoning? All right, again, so to the best of our knowledge, free oxygen is not present on other planets. Therefore, it would be best to look for life forms that have characteristics similar to those of anaerobic archaeal species. What tests or probes would you use to find the kind of life you proposed in Part A, and explain your reasoning? So, to answer this question, you need to make some assumptions. That life on other planets may have evolved similar to life on Earth. This assumption is supported by the Stanley Miller's experiment, the abiotic synthesis experiment, which indicated that key organic compounds present on Earth can be produced under reducing atmospheric conditions. And given that assumption, we could look for some of the compounds we know are common to life, and we could analyze soil for other samples, or other samples for the presence of polymers of carbon, like carbohydrates, proteins, lipids, nucleic acids. So based on that assumption, that the life forms have DNA or RNA as their genetic material, we could use radioactive DNA or RNA probes, making these similar to highly conserved genes in Archaea and other organisms would give you a better chance of finding possible homology and similarities between them. That was a whole lot of talking and not much writing. So, making the assumption that life would follow a similar evolutionary path to that found on Earth, capital E Earth, we could look for compounds we know are common to life. Such as polymers of carbon. Polymers of carbon. Cool. That's our stream. Finishing a little bit early. I'm going to go play in the sun, y'all. I hope you have an opportunity to get outside, drink some water, take care of yourselves. I know midterms are coming up for most of us. So take care of yourselves and hit the books. Thank y'all for tuning in. Don't forget to turn in your worksheets for extra credit.